Hi, my name is Dr.
Gaukhar Baidildinova.
This video is a part of my lectures on evidence-based medicine and epidemiology. The last classification of RCT to discuss is their designs. I found 5 subtypes, and again, they are not completely mutually exclusive. These are matched pairs, treatment withdrawal, parallel, crossover, and factorial. I also created symbols by myself just for fun. After the explanations, I hope it should be clear to you what they mean. Matched pair design. Randomization is not always a practical or ethical method for assigning participants to comparison groups in research studies. For instance, we cannot randomly assign individuals to wear or not wear seatbelts when assessing the effect of seatbelts on motor vehicle crashes. In such situations, alternative study designs, including matched or paired designs, are commonly used. Matched pair design is particularly useful when there are 2 treatment conditions in an experiment, and participants can be grouped into pairs based on similar characteristics to reduce variability in results. This approach is used in both randomized controlled trials, RCTs, and observational studies, though the way it is applied differs. In RCTs, matching can be done before randomization, matched pair randomization. Participants are paired based on key characteristics, e.g., age, sex, disease severity, and then one member of each pair is randomly assigned to the treatment group and the other to the control group. This helps ensure that treatment and control groups are well balanced while still maintaining the benefits of randomization. In observational studies, since there is no randomization, matching is used after groups are naturally formed, e.g., in case control or cohort studies, to reduce confounding. For example, in a study comparing seat-belted and unbelted individuals, researchers might match participants based on factors like age, driving experience, or health status to make fair comparisons. The most obvious example of matched pair design is twin studies. The main advantage of matching is that it minimizes systematic errors associated with baseline group differences, thereby reducing variability and improving the accuracy of group comparisons. This approach is widely used in studies of diseases and health outcomes. However, matching has some limitations. It can be challenging to identify and measure all relevant factors for matching. Overmatching can occur, potentially obscuring true differences between groups. Matching may reduce generalizability if the matched sample is not representative of the broader population. The process can be time-consuming and may reduce the overall sample size. And there is a risk for observation bias. Researchers must carefully consider which factors are most important to match on, balancing the need for control against practical constraints and study goals. Withdrawal design. Participants first receive treatment for a while, and then they are randomized to continue on the study treatment or receive a placebo. Withdrawal of active therapy. That's why there's a fork sign. The withdrawal design helps assess the effectiveness and withdrawal effects of the treatment by comparing those who remain on the treatment to those who are switched to a placebo. An example: an article from Lancet, 2023. In the method section, it says, "This Phase 3, a randomized, double-blind, placebo-controlled, withdrawal, efficacy, and safety trial. Patients with arthritis. First 2 weeks they checked safety and pharmacokinetics. Then, for 12 weeks total, including these 2-week safety trial, all the patients took baricitinib, and then for 32 weeks it was placebo-controlled double-blinded studies. Patients were randomly assigned to receive a placebo or continue receiving baricitinib." I also have 2 paid courses on Udemy. The Critical Assessment of Clinical Studies teaches how to read and critically evaluate clinical studies. It's a very great course. The same as my second course, Scientific Writing, where I tell all the nuances of how to write a scientific article or dissertation. Links are in the description box. Parallel groups. In the case of parallel group randomization, after randomization, each participant remains in the assigned treatment group for the duration of the study. The symbol here is more than transparent: 2 parallel lines. The parallel group design can be applied to many diseases, allowing experiments to be conducted simultaneously in multiple groups, and these groups can be in different locations. The problem is the homogenization of groups. A study from BMJ. Right in the title, it says the pragmatic, parallel group, open-label, Randomized controlled trial. Patients with computer-generated sequences were equally randomized after hospital stratification to those who would go through single or two-stage revision procedures. Crossover design involves participants receiving different treatments sequentially, e.g., receiving a candidate substance in phase 1 and a comparison control drug in phase 2. Each treatment starts at an equivalence point, and each participant's condition is monitored relative to their own condition. I think the badge is clear too. I would compare this design to crossing over as well. The design has certain advantages, such as low variability, because the same patient is in the treatment and control groups, and the possibility of evaluating several treatments. However, there must be sufficient time between the different phases of treatment, a washout period. And this design can only be used in chronic disease research as therapies come one after the other. I looked through PubMed, and most of the articles are in the nutrition field, which makes sense because diet changes is not dangerous in contrast to different medication switch. But I also found studies where different visualization techniques applied for surgery assistance were crossovered, if I can say so, or ophthalmology paper where they gave patients with myopia to try on 3 different lenses in random order. For a closer look, I chose another nutrition article. Here we have patients with the irritable bowel syndrome and 3 types of diets. Diet A is FODMAP, which is an abbreviation for fermentable oligodii, monosaccharides, and polyols, and gluten if you are interested, but it's not a point. B. Gluten. C. Placebo. And patients were randomized to sequences CBA, A, C, B, B, A, C, meaning those who were on CBA first were on placebo, C, then on gluten, B, and then on A, FODMAP. Factorial clinical trials study the effect of more than one treatment. This allows potential interactions between different treatments to be evaluated. If you look carefully at the scheme and the icon, you will realize that it is the same principle as for the combination of heterozygote alleles. For illustration purposes, I chose a reproduction-related article. Here we have a long title. The essence is the following. We have women who took clomiphene citrate. Then some of them stayed on this drug, others switched to gonadotropins, and in each group they tried either intrauterine insemination or standard intercourse.